and by the way, I changed the title, and that's because I'm a rebel. And the second reason is that we have actually a book called with the same title. So Orchestrating Microbiome Analysis with Bioconductor. So welcome, and in this session we go through this miaverse, which is the summarized experiment framework for microbiome analysis. And first 10 minutes, hopefully we have the 40 minutes, let's see. But first 10 minutes I will go through the background, and then after that 20 minutes the demonstration, and hopefully we have still 10 minutes for you to explore the OMA book, the online book, and the material that I showed you during the demonstration. So after this workshop, I hope that I have convinced you to use these tools. And secondly, you should have like basic knowledge on this TreeSummarizedExperiment object and the methods, related methods. And thirdly, or the most importantly, you know where to find the help, and that's the OMA book, the online book. That's the solution for everything. So I start with the question: how many of you are more like experimentalists, performing analysis for wet lab data and that kind of stuff? All right. And how many of you? So the rest of are like more like developers and that kind of. Okay, cool. So Shivang, I have good news for you. It's like by using these methods, you can apply cutting edge tools from Bioconductor. And for developers, I would say that this makes your life easier because you can trust that the data is in this standardized format, and you get bigger audience because Shivang can also use these methods then quite easily. So Bioconductor relies on data containers, especially on a summarized experiment data container, as it is most widely used in different fields, various different fields. However, microbiome field has previously relied on phylogenetic, which is a problem. Why? Well, I'm gonna tell you why. Because summarized experiment, there's a lot of there's a wide variety of fields, and then when they are applying the same data container, they can share methods, share ideas, collaborate, and that's like the core of this Bioconductor. And for instance, if we think about genomics or single cell analysis, there are lots of common things with microbiome. However, we cannot utilize this synergy if we are using phylogenetic. It is like being in a corner right there. And networking, but we are there. Until now, because SummarizedExperiment has extended the microbiome field, we can, in the middle of the dance floor, having great moves, and really like a benefit from the whole Bioconductor, and really like a harness the essence or the soul of Bioconductor collaboration. So now we know that we should use SummarizedExperiment in a microbiome field also. So let's discuss about this SummarizedExperiment data container. So the core of this SummarizedExperiment data container is this abundance tables. So we have there rows and we have columns, and the rows represent features, taxa, bacteria, different bacteria, and then we have columns which represent samples. And for both, we can have metadata. So we can have, for samples, we can have, for example, the patient metadata, what diagnosis each patient have and that kind of stuff. And then in row metadata, we can have information on or the taxonomy table, for instance. So in which taxonomic group this specific bacterium belongs to, or something like that. And in microbiome field, we have this specific data types that we have. We usually want to incorporate in our analysis. And this summarized experiment does not yet include those, but we have extended this or Huang et al. Extended this summarized experiment data container to TreeSummarizedExperiment, which contains these data types that we usually include in our analysis. For instance, we can have those reference sequences there and the phylogenetic tree, which is very important. So phylogeny, the hierarchy between bacteria. So we can have that in row tree. And additionally, we can have also a hierarchy tree for samples, so column tree. And that can, for instance, represent if patients are like relatives or something like that. So now we have the TreeSummarizedExperiment, SummarizedExperiment data container for microbiome analysis. So of course we need methods to analyze the data in that format. And then we develop these MIA packages. Can you actually see that? A little bit small. Those MIA packages, so microbiome analysis packages. And those MIA and MIaVis packages, they contain those common methods for data analysis and visualization. And then we have also MIaSim and MIaTime packages, which have those data simulation and time series stuff. And then those independent developers have developed this their own methods, and they are now supporting this TreeSummarizedExperiment. So we have broad tools now in this miaverse ecosystem because of this. And to put this like a little nice package, we have wrote this Orchestrating Microbiome Analysis online book. So it aims to like combine best practices and also the information on microbiome data science, and it is developed with a collaborative effort. So any kind of contribution is welcome there. Yes. So hopefully this sounds quite tasty. So we are ready to hands-on session. So yeah. So we in this hands-on we follow the introductory workflow from OMA, the online book, and you have also the virtual machine there in Bioconductor. So I will let you have some time to start your engines, and might be good time to if you have questions. So please feel free. No questions. Well, then I can show you the where to find the virtual machine. So it's there in Bioconductor. There's this package demo MIA. I have already it here, but then the introductory workflow, you can get access. It's actually there, like this. So I can show you the OMA book a little bit. So as I said, it's the online book, and it has lots of information on microbiome data science. There's an introduction, and then there's different kind of topics here which are related to microbiome stuff. Yeah. So I assume that you are ready to follow me on this workshop then. Yes. So, yeah, so in this workflow, we demonstrate how to utilize this miaverse framework, and we cover these common topics like transformation and agglomeration and stuff like that. And then we do the basic exploration step, and then hopefully we have time to analyze the alpha and beta diversities. So first of all, we have to download some packages. There's Mia and miaverse, and then a scatter package. And you can load the packages with this script. This is just for like if you do not have those on your own computer, then this also installs those packages. So let's do that. So nice, no errors, so we can continue. So in this demonstration, we analyzed the data, Tengeler data, and they studied microbiome between controls and ADHD. So they tried to understand if there's any differences in their microbiome, those patients. And this demonstration data set you can find from MIA, and then in OMA you can also you can find more information. We have also packages for fetching data from different databases, so you can find more information from there. And first of all, we see that this data set is TreeSummarizedExperiment object. As I mentioned you, this TreeSummarizedExperiment, it is a summarized experiment object. You can see this. See, I didn't lie. And you can, in fact, convert it to summarize the experiment. That's just a stupid way to do because it just loses some data. So let's not do that. To analyze the data in this TreeSummarizedExperiment or any kind of format, of course, we have to be like comfortable with the data format. So let's first learn how to subset and access the data. So as I already mentioned, this TreeSummarizedExperiment, it can have or it has rows and it has columns. So the subsetting works similarly to any kind of matrix objects. Take 10 first rows, and we can see that now the dimension for rows is 10. So we have subsetted the rows, and then we can subset the columns by specifying the. For instance, we can take the 5th sample. Why not? Let's take that. And we can see that now the dimensions are 10 and 1. To make it like more like well, that was just arbitrary, but then we can, for instance, in this workflow, we can subset the data based on this cohort, so we can take the cohort information from column metadata, like not like that. Oh yeah, but this, and we can see that now there's information for every sample on which column cohort they are included, and then we can take only those that are in cohort 2. We can get the logical vector, and then we can finally subset the data, and we can see that now we have now 10 samples. Then I promised you to show how to access the data, so we can access the abundance table. It is in essay slot, as you can see from this TreeSummarizedExperiment object. It contains different slots. So here the counts table is the abundance table. So we can access that counts table by first specifying this assay function and then specifying the abundance table that we want to fetch. And we can see that now it includes those counts. Then we can access the column metadata with call data function. And we can see that it includes those patient information and diagnosis and cohort and stuff like that. And now we can actually see that there's only now cohort 2 because we subsetted. So, and finally, we can access the taxonomy table, and you can see that now there's a different taxonomy ranks, and here rows represent the different bacteria, and it shows that in which taxonomy group this certain bacteria belongs to. All right, is this now clear? I'm following good. Then common thing in microbiome workflows is agglomerating data. So we summarize the data in a specific taxonomy rank, for example, phylum taxonomy rank. So MIA includes that function, and we can easily apply that. And we can now see that the dimensions has changed. So we have five rows, 5 different rows, which is something to do. It is something that you should like. What's the word? I lost the word. But that is good thing. Sorry about my English. But we can check the taxonomy table now, and that is also a good thing. So it is like something to like wait for, because now we can see that there's all the like from phylum and the ranks after that they don't have any information because we have combined the data to this phylum level. So it is something to expect. And now we can actually check also the abundance table, and you already know how to do that, but let's do that together. So we can see now that there's those bigger values compared to the last essay. So it has summarized the counts. Then we have to, in microbial workflows, we have to apply some transformations, and MIA includes this transform assay function, which has this, in addition to this like a basic relative abundance methods or transformations, it also includes those more microbiome specific transformations. But let's apply relative abundance, and then we can now we can see that there's a additional table, relative abundance table, and similarly to counts table, we can access the relative abundance table. And now we can see that they are in scale from 0 to 1, so the transformation was successful. Cool. So now we have now we know how to subset the data, and then we know how to access the data. Then we know how to apply this agglomeration transformation, which are part of this pre-processing in microbial workflows. So then we can continue with our workflow by summarizing the composition. So that's something that is very commonly done in if you look those microbiome papers. There's like every paper has this compositional bar plot. So this function is in miaverse package, and we can see from, oh yeah, it goes there in the other panel. Yeah, let's do it like this. Yeah, now we can see. So it creates this kind of plot, and these are actually all these plots that are coming from miaverse or this framework. They are GG plot objects, so we can actually quite easily modify them. Yeah, it's a nice plot. Yeah, so that confirms that it's a nice plot. So we can see that this like demonstrates that you can actually see here better. So you can see that it summarized the compositional or the relative abundances of this phylum level, and Bacteroidetes is it seems that it's the most common bacteria there, and then also that when it's relatively low its abundance, then the Firmicutes is the abundance is higher. I'm not a biologist, so I don't know what that means, but that's just something that we can see from the plot. Then we can analyze this alpha diversity. So this also common step in microbiome workflows. So we're in this alpha diversity, we measure how diverse is the microbial community in a sample. So for instance, if we have lots of different bacteria and they have equal abundances, it means that it has a great diversity. But if there's like just a couple of species and they have very dominant species, then the diversity is low. So we have this function add alpha, which calculates these diversity indices, and we have different indices there, but let's calculate this fate index because it incorporates the phylogeny into the calculation.
Yes, from which package is add alpha?
Add alpha is in MIA package. Yeah. So what it does, it calculates the values and adds them at alpha, so it adds them to the object, and we can find the calculated values from call data. So let's see. Yeah, so they are there in the fate column, and then we can visualize the values, and this is great example of this sharing methods between this summarized experiment ecosystem, because this plot call data function comes from scatter package, which is a single cell analysis package. So we can utilize that package. And let's in x-axis, let's plot this diagnosis, and then in y-axis, let's plot this faith index. So, well, let's see this here. It's bigger. So, can someone explain what we see here? Well, yeah, there's no difference, so we don't get to nature with this finding. But this diversity does not tell everything. So there's in workflow there's a beta diversity calculation step, but yeah, I will let you to do that. So it's your time to shine, and let's continue with that. I think I have like a 10 minutes still. So.
Oh yeah, it was supposed to be this, and so yeah.
Or do I have 10 minutes?
Double check. I cannot memorize the schedule. It was supposed to be 9:45, and oh yeah, 10 minutes. 10, even 15 if we said it. We did it. That's weird. So it says the next session starts at 9:45. So you're still on time now. And yeah, we were saying that we delay by 10 minutes, so you could even go to 55.
Okay, well,
unless you want to give people time to essentially move into the.
Yeah, that's cool. Let's
aim for 10.
Yeah, 10 minutes, and you can continue with the workflow, and then the additional steps. There's a differential abundance step that you can follow from the links, and you can explore the OMA book, and we can discuss if you have some questions, and then I can summarize in yeah, seems that they are ending, so we can also wrap this up. Yeah, well, first of all, thanks for joining. It was nice. But yeah, about the final words. This is the summarized experiment framework for microbiome analysis. And remember that you want to be in the dance floor and not in the corner. So that's why we are using summarized experiment. Then there's the online book OMA. You can find information from there. And also, if you are interested to contribute, please you can create a pull request, or let's talk in Slack or something like that. Because the idea is that it's like not just me and Leo, but like the whole Bioconductor communities collaborating and creating nice stuff. And yeah, if you have questions, future questions, you can find information from OMA. Here's somewhere also my email and stuff. And Leo, do you have any final words?
No, not really. It's nice to have you all here, and I think if you want stickers, Tuomas can give you some.
Yeah, that was my final point, because as we are from the land of Santa Claus, so we have to give you some presents. So we have actually T-shirts here.
Oh.
So we have also stickers. So if you prefer those.
Maybe one point that Tuomas didn't highlight many times, at least, is the interoperability with like metabolomics, proteomics, and cells. Yeah,